Thanks for the introduction, and I'm so glad to be here and share with you our recent work. Still working on the atlas. From jaws, you can see different atlas from different species. Now I go back to the human brain, and using the in vivo and non-invasive neuroimaging technology, especially in the diffusion data, to create a new atlas. We named it Brainnetome Atlas. And this atlas was published last year, and today I will show you one part of this atlas, the Orbitofrontal Cortex. I'll go to more details about this region and this atlas, how to use this atlas to explore your questions. So the title of my topic is uncovering the connectional architecture. Firstly, we using diffusion to map all connectograms of each part of the orbitofrontal cortex. We identify 13 subregions in this area. And also we're using other ways to decoding the structural and functional relationship and organization of this region. From beginning of the story, I want to show this great atlas, Brainnetome Atlas. Why it's great? It's not only show us the brain areas, but it gives us some functional definition of each area. If you mention the area 1, you mean this is the sensory area. Area 44 is language area. So the atlas will not only give us where the region it is, it should give us more information like the functional. Also, there are lots of other information like what is the connection, what is the development, the four-dimensional information from this atlas. So Brainnetome Atlas is very important for us, but it's a little bit rough. Just have 44 areas in the human brain, and it's just based on one specimen, postmortem brain. So you neglect lots of individual variants. And so this is the basic idea. This is a lab from Vogt. The Brainnetome mentor is here. They're using two ways actually. Two histology ways, staining techniques to got the atlas. One is the myelin architecture, the other is the subtle architecture. The subtle architecture means the distribution of different cells between the three different layers in the brain. They're using these criteria to identify the brain areas. So this is the main, this is three examples of including the motor cortex, sensory cortex, and other high functional high order of areas in here. Just using these criteria to identify the boundaries of the different brain regions. Literally, using this information to remap to using the image data into the standard space like the MR space. So from that atlas, there existed a lot of atlas. Most of the early atlas are based on the histology ways, include different criteria, like including subtle architecture, mallow architecture, chemical architecture. Also, recently they're using receptor distribution in the different layers in the brain to identify the boundaries of the different brain regions. With the development of the neuroimaging, especially the MRI, we can got the in vivo data, including the structure. We can got the SOSA and gyri. Also, we can using other modalities like the diffusion data to got the anatomical connectivities. Also, using the functional data, we can got a statistic analysis in the resting state, got the functional connectivity. Also, we can use different tasks of FMR using to map the functional region. Our atlas, most early atlas of the imaging data is like the free surface atlas, AL atlas, Harvard-Oxford atlas. They're just based on the micro-anatomical landmarks, the SOSA or gyri to identify the areas. So the problem is there. There are later correspondence between the anatomical SOSA and gyri with the function of the brain. So our basic idea is using the connectivities to identify the brain areas, because we know there's a very close relationship between the functions and the connections. So here is, exists a lot of milestones in the brain atlas history. We We hope our atlas could be one of them in the history. So I will go to more details about how we do the brain postulation, how we created the atlas here. So firstly, first thing is to create an atlas is to identify the boundaries. So we use the connectivity profiles to identify the boundaries and then got the areas. So I want to first give a definition. What makes a brain area distinct? The brain areas are functionally unique subdivisions distinguished from one another by three main criteria. One is the architecture. It's including different types of the architecture. Using in vivo data, some groups using using T1 divide T2 got the Mellon map in vivo. Also, we can measure the thickness or area of the brain region. It's also other measurements. This could be the architecture, but I think the basic of the architecture is using the histology ways, including like the subtle architecture. Also, we know there's very close relationship. If you want to know more details, you can read this review paper. They have close relationship between the connectivity and the shuttle architecture. The second is input and output connections to identify to give the functional distinct region definition. This figure shows the COCO map that collected all the existing track trace and data in that database. And the participant got give a review paper, give a definition of the connectivity fingerprints. It means that each individual region should have a very distinct fingerprints. If you use the track tracing data, you can get input fingerprints or output fingerprints. But when we use the diffusion data, we got just one fingerprint because the data can't give the in vivo diffusion data can't give us the directions of the fiber pathways. And finally is the patterns of gene expression, especially from the development view. Two examples. One is from the animal studies. If you knock out some genes, the brain can't be separately anteriorly or posteriorly. In other genes, the brain can't go to the medial and lateral parts. It means when we finally got the mosaic of the brain areas, there are lots of genes plays very important roles in this processing. Recently, published one paper in Nature Neuroscience. They compared the functional connectivity pattern with the gene expression data from the Allen Brain Institute. They find very close relationship between the two informations, the gene expression and the connectivity. So, in summary, there are four main criteria to definition the brain area. One is the function, but the function is a little bit confused for you to identify brain area. You can't let a participant subject to do all the tasks in your study. So if you're using function to identify the boundary, it's no ending work. The other two is the architecture, usually based on the histology ways. And the third one is the topology. Topography. Topography usually works on the primary cortex, like the sensory motor cortex, visual cortex, and auditory cortex. Like the auditory cortex, there are different frequency of the voice can activate in different parts of the auditory cortex. This is called topography. And the fourth one is connectivity. We think the connectivity maybe plays a fundamental role in this among these four criteria. For example, we do some postulation using connectivity. We can identify some functional areas, especially in the Brahman area 44 and 45. We got ventral and dorsal parts of the 44 and anterior and the posterior part of the 45. This is very similar to the recent receptor distribution from the Ulysses group. They've also found these fine-grained puzzles, and it can be resembled to the function of this region. The other example why we don't use in the histology, the architecture, like cerebellum. All the cerebellar cortex have unique architecture. What can define the different functions of the cerebellum cortex? It depends on the connections with the cerebral cortex. It depends on the connections. And the other example is area 17 and 14 is a primary visual cortex. Usually in the subtle architecture, they said this area 17 have a thick layer 4, but you can see if they have a thick layer 4, the function of this area is proceeding in visual processing. We think that the connection between the visual system determines the function of the area 17. So we think that this is the fundamental thing. The connectivity determines the function of the brain region. So we using this information to do our atlas construction. We have this paradigm. Firstly, you take the temporal pole, for example. You got 1,000 voxels in this area. You can using different data sets to map the anatomical, functional connections of this. Each voxel in the brain region. You can two matrix, the connectivity matrix. Also, you can got the similarity matrix using clustering algorithms, different kinds of clustering algorithms to do the postulation. That one problem here, how to define the final class numbers. Here we're using the stability across the datasets to get the final solutions. So maybe it's not the most fine-grained puzzle, but it's the stability across according to the datasets you used. So the last 6 years, we got several regions to published on the journals. Each paper we describe very details of each region in these areas. When finally we got this atlas, the human Brainnetome atlas, got 246 sub-regions across the whole brain, 210 in the cortex. And that's a little bit rough in the subcortical area. I was still working on that. We just got 66 in the subcortical areas. More importantly, we got the connectivity profiles of each subregion to the rest of the brain areas. So we go back to the orbitofrontal cortex for more details of how we build this atlas. Firstly, why we do focus on this region? Here, 3 subjects. You can see that if you, according to the circuits on gyres, they're very huge individual variance. You can't just using this micro landmarks to identify the subregion in the orbitofrontal cortex. Different subjects have give you different results there. If you want to use the histology ways, there are lots of choice you can choose. From 3 to 17, different histology ways gives you different results. So there's no final atlas of the orbitofrontal cortex now. There also exist lots of in vivo studies, but most of them just based on SACA and gyre from first, like the free stuff for AL. Our collaborator from Tianjin using the structure covariance give you orbitofrontal cortex. It's just a group postulation. You have just a female group and a male group, and just have 5 solutions here. So still rough in this region. From the functional views, there exist a lot of studies. They show that different functions can activate, like here, the punishment or winning can activate the anterior part and the posterior part. Also, the different values of the reward can activate the middle part and the lateral part. Also, actually, there's lots of meta-analysis, different tasks to activate the different subregions. This shows us this orbitofrontal cortex. So they have a hierarchical postulation organization, and they have a very fine-grained subregions in this area. Also, we can got the puzzles here, but we still miss the connectivity architecture of the orbitofrontal cortex. All the literatures we used, we researched in the PubMed is based on the macaque data. That's just the generalization of the monkey connections to the human brain. But we know that there are very huge cross-space difference there. So the purpose of this orbitofrontal atlas, firstly, they got a fine-grained puzzles and maybe a hierarchical organization here. The second one is we want to map the connectional architecture of the human orbitofrontal cortex. And finally, we're using the Brainnetome database to do the decoding of what's the function of each orbitofrontal subregion play in the different tasks. So we're using datasets from the Human Connectome project because they solve the problems, the signal distortion in this area. In the EPI sequence, we from our usually data, you always see the ghost or signal loss in this region, but this project gives us very good data sets here. And also they have very high resolution diffusion data. It's 1.25 millimeter isotropic. Firstly, how to draw the mask of the orbitofrontal cortex. We are using the free surfer to individually identify each circus and gyra in the orbitofrontal cortex. So we collect these ROIs into one mask. We name it orbitofrontal cortex for each subject. Then we put it into the connectivity-based postulation pipelines using the index. This index shows the stability across the subject, the dataset. They have 3 solutions, 3, 6, and 15, and gives us is the stable solutions in this area. This is the postulations. You can got 3 solutions here and 6 and more fine-grained 15. I will go to the details about the 15 solutions. Using our ways, you can identify the media parts area, area 10, area 11, area 13. In the middle is the area 13, and the later part is area 14, 6, and 12, different subregions. So totally they got 15 solutions, but 2 regions is not belong to the orbitofrontal cortex. One is the anterior 14-6 part, and the other is the red one is anterior inferior frontal sulcus. These 2 regions is belong to the lateral prefrontal cortex. So totally there are 13 is orbitofrontal cortex in our study. And this, we are not only gives the maximum probability map, we also got this individual variance atlas, the probabilistic maps of each subregion. Also, give us same story about in the left side of the orbitofrontal cortex. But here we got 16 subregions. That just separate this middle part of the middle 14 into 2 subregions. So also this, I think this is a hierarchical atlas. That's a symmetry between these 2 hemispheres. Got 3 in this region and 6 very symmetric in the postulation results. So then, once we got the puzzles in the orbitofrontal cortex, we want to map what about the connectogram or the connectivity profiles. This figure shows the major 5 pathways from each subregion to the rest of the brain. And we're using the quantitative ways to do a hierarchical clustering. They give 2 disciplines here. They show this just using the 15 fiber pathways to do the analysis. They show very similar to if went to the first tab, we would just direct postulate this region in the 3 and 6. These 15 areas give us a very hierarchical organization in the medial orbitofrontal cortex and the central parts and the inferior lateral part, the complex there. Finally, we mapped this 15 subregions, the connectogram, to the rest of the brain, to the striatum, the thalamus, and this sensory motor limbic cortex. You can see these 15 subregions so very distinct connectivity to the rest of the brain. So this is the connected architecture of the orbitofrontal cortex. Also, we use the resting state to map the functional networks. What is this 15 subregions involved in different functional networks? We cooperate with the Jülich group, Simon Ackhoff. They give us the Brainnetome database. They collected 15 domains, behavior domains in their database. We use that database to decoding what is the function in these 15 subregions. So the 15 subregions, different domains can activate each subregion. And also we got the paradigm analysis. What kind of paradigm can be activated in subregion? Example, here the reward paradigm. Almost activate each part of the orbitofrontal cortex, except for the anterior 46 and the inferior frontal sulcus. These two regions cannot be activated in the reward activity. So maybe we can go to more details of what the function of each subregion plays in these figures. All this data will be online. You can identify where it is and what the connection is and what the function is. And finally, we want to give a summary and a few suggestions. Here we presented a very detailed postulation and also a hierarchical postulation in the orbitofrontal cortex. We use the diffusion data to remap the connectivity architecture of each part in the brain, and we can use this compared to different, maybe crowding different spaces. We uncover the functional heterogeneity of the orbitofrontal cortex. What the next maybe to understand the relationship between the connected unit or OFC subregions with patterns of a gene expression, we can compare with using the Allen brain datasets, what kind of the difference there. And also, we can review the evolutionary specialization of the orbitofrontal cortex connections in macaque and the humans. Now we have collected lots of post-mortem and in vivo macaque brains. We can just use the diffusion to compare the cross-space comparison. And finally, to remap the individual variability of the connections and its dysfunctions in different types of disorders related to this brain region. And finally, I want to thank all the collaborators in our institute and the Shandong University and the Jülich group and Sichuan University. Finally, thank you very much.
Thank you for the great talk. As you have mentioned, there are multiple atlases existing here, some based on resting state alone, some based on multimodal imaging, resting state DTI, and sub-architecture and clusters using human connectome project. So my question is, what's your comments on the usage of these atlases? So if you have a specific project, what atlas, what's your, how to select the atlas to use? Second question is, in your definition of the whole brain Brainnetome atlas, you seem to first start with some big areas and then put together a whole brain. So how do you define the boundary of the big areas? And then is that manually done or what? So that's my question.
Yeah, very good question. The first question is different modalities. As I talk and say that you can, firstly you should find the more fundamental things. If you put lots of modalities together, then maybe they give you different stories. You can't have final solutions. I think the connectivity maybe plays very fundamental roles. Actually, for the connectivity, you can got different modalities: the anatomical, functional, maybe co-activation in different tasks. We have that second version of the atlas. We are working on that using different modalities of connectivity. We published several papers, like the superior from parietal lobe. We got very similar across these three modalities, 5 solutions in this area. How to use these different existing atlas? Maybe that's one. I think we go 2 ways. We just using one modalities. The other way go the multimodal postulation, like the Nature paper. But for nowadays, maybe within 10 years, we're just using, if you do the group analysis, using the volume-based registration, according to the main circuits and drivers to do the registration. So now we think our atlas, the Brainnetome atlas, is based the volume-based atlas, and in the MAR standard space, maybe it's useful now. But later we are still working on different registrations using other information to do the registration. And the other solution is our atlas can be built individually. Actually, we do the postulation for each individual. Now we tested the reproductive of this method, whether it can be worked on each individual. Actually, when you see some papers using the resting state functional connectivity to do the postulation, if you collect different data in different time points, they give you even at the network skills, the same subject will give you different solution at different time points. And second question is, second question is, the big area. Big area. Okay. So this why because we still using the volume-based registration according to the main circuit and drivers. So we identified, we collected using the free surf atlas to the initial population. We identified 20 subregions. We think that the main circuits maybe have little individual variance between different subjects. So we use that as the first initial population. The second one, if you put the whole brain in the pipeline, you don't even know how many clusters in this brain. We should put some previous knowledges on this region. So,
it's interesting you have the human connectome, you tested the Brainnetome and the human connectome data. They have an approach there which comes close in vivo to cytoarchitectonics, which is to look at mirroring by dividing T2 by T1. Have you done a comparison of this with the Brainnetome, which is based on diffusion weighted?
Yeah, we didn't compare with T1 divide T2. We compared with the subtle architecture atlas, like the U-brain. We found a very close relationship with that atlas because we cooperate very closely with that group, group. But we will go to area 10, area 35. If you just use subtle architecture, they can't give you the fine-grained puzzles there because they just have a very homogeneous subtle architecture in these areas. But later for the other modalities, we can go on our work. If we separately use different modalities, we'll see they have similar boundaries or different boundaries. Maybe they can create different stories there. I'm not sure. We are still working on that.
Lingzhong, you find left and right differences or principally similar? I mean, if they are similar, it increases your confidence that you're actually picking something real. But on the other hand, there might be expected some differences in connectivity in language areas, for example, that are not so they're not present in the right hemisphere. While I have the microphone, the variability that you mentioned, you wouldn't expect that you will have gross difference between the individuals. You will have, of course, differences in the pattern of folding, but the underlying cytoarchitecture should be of similar. There would always be the areas in the right order, in the same order, never jumping around. And the real estate that they have should be within limits similar. And finally, what you said that there are huge differences, interspecies differences. Have you seen those? Because Petridis and Pandya didn't find any. And we think, certainly with risks and marmoset that we did in parallel, they are there, the areas and roughly in the same areal size.
Yeah, to answer the question, and it's a very useful to guide our next analysis. We didn't compare the asymmetric in the brain. Actually, we use the asymmetric as an index to define the fine numbers in the whole brain atlas. Not one is based on stability. The other is asymmetric between the two, symmetric between the two hemispheres. So we got the same class numbers in two hemispheres. But here, for the orbitofrontal cortex, you can see that the right side got 15 is the most stability, and the other side is got 16 is the most stability. Just to separate one region into two regions. So I think there is a limitation here. We're just using the stability to define the final solutions. But later we can bring some other index to guide how to finish this work. But I think there's still ways to go. About asymmetry, maybe next we will do some studies to map the size and the connectivities and the functions of the two regions. So it's very good comments. We can keep working on it.
Thank you for the next talk. So I have a question about if we look at the details of the fiber connection, we can find that most of the fiber connections are concentrated on the gyro regions, not the sulcal regions. So if we use the fiber connection as the features to do the pacillation or to build up the atlas, so we can see that most of the identified subregions are on the gyro regions, not the sulcal regions. So do you have any comments or insights? So how we can find the atlas or the subregions of the sulcal regions based on the fiber connectivity features?
Yes, very good question. Last week we were still discussing about this topic. We know that there may be some false positive in the circus, the bottom circus of the fiber tracking. We did some work on like the posterior superior temporal circuits. We're using the diffusion data, using the function data, using the task, the Brainnetome database. They give us a very similar population that got the anterior and posterior part. And how to solve that problem? Maybe if you have a high resolution, maybe I can show you actually what's the fibers from the bottom of the circus. Maybe using other ways, using the invasive ways, do the tracing, the track tracing, using the monkey data, because monkeys to have the circus. Because we have a lot of monkeys there, we will work on that because it's more fundamental for the basic for the DTI-based track tracing. You can. I can't give a clear answer to that question. We will work on that. Yeah.
I have a follow-up question along those lines. Given that limitation of DTI-based tractography for looking at the white matter, this gyral bias and things like that, following up on the myeloarchitecture studies, the T1-T2 ratio, as well as I understand Glasser uses cortical thickness for things analogous to cytoarchitectural parcellation of the cerebral cortex. One thing that I haven't seen the human connectome group do that it seems like you're very well poised to do is to look at diffusion anisotropy or other diffusion metrics within the cerebral cortical gray matter. It's been shown that this 1.25 millimeter isotropic resolution does reveal some interesting differences between different cortical regions within the gray matter. I think that that's only going to become a richer source of information as our ability to get higher and higher image resolution comes along. So I'm wondering if you have any thoughts for using that as a source of information and maybe as a comment to advocate to look for that from the diffusion MRI data.
I Actually, one thing I should correct it. The HCP group, Oxford University belong to that project. Actually, they did a lot of work on the diffusion-based postulation in the brain region, but not in the Nature paper. Actually, you can see the literature, lots of postulation results based on diffusion from the Oxford group, the FSL group. The second one is the comments. If we can get a very high resolution dataset. Yes, we have 9.4 Tesla MR scanner in our lab. We have collected some datasets from the post-mortem brain, both the human and the monkey brain. If we have the high resolution, also we have other techniques like the PLI to get a very high resolution. We can see what's the golden standard of the fibers from the bottom of the circuit, where it goes. It goes to the banks of the circuits or just go into the fiber, white matter, the main fibers. I think that this may be our next step. We can collect the data and release that data to help guys to find some new algorithms to do the fiber tracking. Because first thing, we should have that data set.
Thanks very much.
Thank you.